Cellular respiration is a process by which cells acquire energy by breaking down nutrient molecules produced by photosynthesizers. Its very name implies that cellular respiration requires oxygen and gives off carbon dioxide. In fact, it is the reason any animal such as an ocelot or human breathes, and why plants also require a supply of oxygen. Most often, cellular respiration involves the complete breakdown of glucose to carbon dioxide and water. So glucose plus oxygen yields carbon dioxide and water plus energy. This equation points out that cellular respiration is an oxidation-reduction reaction. Recall that oxidation is the loss of electrons and reduction is the gain of electrons. Therefore, glucose has been oxidized and oxygen has been reduced. But remember that a hydrogen atom consists of a hydrogen ion plus an electron. Therefore, when hydrogen atoms are removed from glucose, so are electrons. And similarly, when hydrogen atoms are added to oxygen, so are electrons. Glucose is a high-energy molecule and it breaks down products, carbon dioxide and water, are low-energy molecules. Therefore, as the equation shows, energy is released. This is the energy that would be used to produce ATP molecules. The cell carries out cellular respiration in order to build up ATP molecules. The pathways of cellular respiration allow the energy within a glucose molecule to be released slowly so that ATP can be produced gradually. Cells will lose a tremendous amount of energy if glucose breakdown occurred all at once. Much energy would become a non-usable heat. The step-by-step breakdown of glucose to carbon dioxide and water usually realizes a maximum yield of 36 to 38 ATP molecules. Dependent on conditions to be discussed later, the energy in these ATP molecules was equivalent to about 39% of the energy that was available in glucose. This conversion is more efficient than many others. For example, only between 20% and 30% of the energy within gasoline is converted to the motion of a car. NAD and FAD. Cellular respiration involves many individual metabolic reactions, each one catalyzed by its own enzyme. Enzymes of particular significance are those that use NAD, a coenzyme of oxidation reduction, sometimes called a redox coenzyme. When a metabolite is oxidized, NAD accepts 2 electrons plus a hydrogen ion, and NADH results. The electrons received by NAD are high-energy electrons that are usually carried to the electron transport chain. NAD plus 2E plus H plus yields NADH. NAD can oxidize a metabolite by accepting electrons and can reduce a metabolite by giving up electrons. Only a small amount of NAD need to be present in a cell because each NAD molecule is used over and over again. FAD, another coenzyme of oxidation reduction, is sometimes used instead of NAD. FAD accepts 2 electrons and 2 hydrogen ions to become FADH2. So basically, cellular respiration gives off carbon dioxide and it requires oxygen. So that's what we need, oxygen. So, okay. And then NAD and FAD. So NAD is a redox coenzyme and accepts 2 electrons and 1 hydrogen ion. And FAD accepts 2 electrons and 2 hydrogen ions.
Phases of cellular respiration. Cellular respiration involves 4 phases: glycolysis, the preparatory reaction, the circuit acid cycle, and the electron transport chain. Glycolysis takes place outside the mitochondria and does not require the presence of oxygen. Therefore, glycolysis is anaerobic. The other phases of cellular respiration takes place inside the mitochondria, where oxygen is the final acceptor of electron. Because they require oxygen, these phases are called aerobic. During these phases, notice where CO2 and H2O the end products of cellular respiration and ATP are produced. Glycolysis is the breakdown of glucose of two molecules of pyruvate. Oxidation results in NADH and provides enough energy for the net gain of two ATP molecules. The preparatory reaction takes place in the matrix in the mitochondria. Pyruvate is broken down to a 2-carbon C2 acetyl group, and CO2 is released. Since glycolysis ends with two molecules of pyruvate, the pre-reaction occurs twice per glucose molecule. The circuit acid cycle also takes place in the matrix of the mitochondria. This oxidation occurs, NADH and FADH2 results, and more CO2 is released. The circuit and cycle is able to produce 1 ATP per turn. Because two acetyl groups enter the cycle per glucose molecule, the cycle turns twice. The electron transport chain, ETC, is a series of carriers on the cristae of the mitochondria. NADH and FADH2 give up electrons to the chain. Energy is released and captured as the electron moves up from higher energy to a lower energy state. Later, this energy will be used for the production of ATP by chemolysis. After oxygen receives electron, it combines with hydrogen ions, H+, and becomes water, H2O. Pyruvate, the end product of glycolysis, is the pivotal metabolite. Further treatment is dependent on whether oxygen is available. If oxygen is available, pyruvate enters a mitochondria and is broken down completely by CO2 and H2O. If oxygen is not available, pyruvate is further metabolized by the cytoplasm by the anaerobic process called fermentation. Fermentation results in a net gain of only 2 AT per glucose molecule. Ready? On page 135, it talks about the 4 phases of cellular respiration. First is glycolysis, second is the preparatory reaction, third is the circuit acid cycle, and fourth is electron transport chain, or ETC. Okay. At the end point of the phases of cellular respiration, there's the end product called pyruvate. If there's oxygen is available, pyruvate enters the mitochondria and breaks down CO2 and H2O. If there's no oxygen, pyruvate process fermentation. Fermentation is the net gain of only 2 AT per glucose molecule.